All right. I just want to take a couple moments to review what we had actually done yesterday. Mr. Wagner had actually ran through what we wanted to cover yesterday, but I don't think the camera roll actually recorded everything. So I'm just going to go through fairly quickly what we had actually covered yesterday. Okay. And this is probably, or perhaps one page we'll cover when you get back from your trip out west. And I think we have all this content. This is actually posted on the Science Channel. It's just this last page that I think is missing. Okay? And it's some terminology that we've used before. And what I want you to think about, we'll be utilizing this vocabulary that when we look at an organism's genotype, it's the actual alleles that make up each trait. The phenotype is something that you can actually observe on its physical appearance, whether it's coat color or texture or pod shape or plant height. Be traits like that that you can actually physically see on the outside. So we relate the phenotype as an organism's physical appearance. And then homozygous just refers to the alleles. They're the same. They can be two capital letters, they can be two lowercase letters. So it's homozygous dominant or homozygous recessive. And then finally, heterozygous would be an allele of each trait, a capital letter and a lowercase letter. Okay, so I think maybe that part was on the presentation, but the actual problem solving was not. So just for a brief moment, I just want to go through a couple of problems that for some reason the camera shut off and did not record those. Okay, so what we see here, this is what Mr. Wagner had gone through. And I'll just give you a brief opportunity, you can follow along. Okay, it says a scientist is conducting an experiment for pea plant height. In his experiment, he crosses a heterozygous plant with a homozygous recessive plant. What is the genotype and phenotype of the offspring? So every time that you approach these problems, I think the purposes that we want to approach these is we want you to make a key, okay? And in that key, you are illustrating what traits you actually have, okay? So here, we're talking about pea plant height, okay? And in this case, tall is dominant over short. So this tall allele, we just put capital T, that meaning tall, okay? And then a lowercase t, the short, this would be for the short allele. And the reason that that's significant is the only way that a plant can be short is if both of these alleles are recessive. So here we're using some of that vocabulary we had just illustrated on the presentation before. So as the problem goes on to state, we have a heterozygous plant, okay? And then we've got a homozygous recessive plant, okay? So for the trait of plant height, and we know that tall is dominant over short. So in this case, he does what I would guess is a type of cross-pollination, okay? Where he's taking the pollen from one plant and placing it on another. That would be cross-pollination. So in this case, a heterozygous plant. So what does that mean for our alleles? That means they're not the same, so they have to be different. So in this case, we have a capital T and a lowercase t, okay? And then it says homozygous recessive. So in that case, the two alleles, they're the same because it says homozygous and then they're recessive. So it would be two lowercase t's. One of the things that's going to happen that as we move forward is going to state, it might not even say that, it could possibly say a heterozygous plant with a short plant. We don't have to tell you what the alleles are because the only way that that outcome could be short is if it's homozygous recessive, okay? So in this case, this is what we call the P1 generation or parental generation. It's just where we start with that first cross. And something that you are a little more familiar with is these Punnett squares. You possibly did those in life science. And if you're in one of Mr. Williamson's ag classes, you might do that in your ag class as well. And like I said, I've always done, if it's on the left side, I just put it in the left over here. So what we do is, as we look at this, we have a capital T and a lowercase t. And then on the right side, we just put it across the top. So in this process, then we just fill out our 4 boxes here. We just go across and down. We've done this before, but it's just a quick review for you. Okay, so capital T, lowercase t, same thing here, capital T and lowercase t, because we're going across, and for the most part, about 95% of the time, you always put the dominant trait first. That might not be the case when you do dihybrid crosses, but that's for another time and another place. Okay, so then what we see here, I can see unfortunately there's a glare. It looks like it's about right there. So we have capital T, lowercase t, capital T, lowercase t. Now we're doing the same thing, going in this manner. We have 2 lowercase t's then. So what we do now for our phenotype, this is not parental, this is our phenotype, is just showing what our genetic makeup is. So we pick a box, we just do this one for our phenotype. Darn, I got that wrong. I'm glad I caught that. Our genotype, our genetic makeup. Okay, so we just pick a box. We have a capital T and a lowercase t. Okay, and then what we want to do is keep track of how many of them are there. Well, we said mark a box. There's one. Is there another one like it? Yes, there's one right next to it. So there's 2. So we have 2 of those. Then, in this case, we see another genetic makeup. We've got 2 lowercase t boxes. Here's one, and we see that there's another one here. When you mark these off, then chances are you won't get those confused, or you won't do a box twice. It's probably not such a big deal when we're talking about these monohybrid crosses with one trait, but once you do these dihybrid crosses where there's 16 boxes, it gets to be a little more tricky. Okay, so in this case, we've got 2 of these as well. Okay, so our phenotype then is what we would actually observe. Okay, and remember, this is where your key comes in handy. When that dominant trait is present, in other words, when a capital T is there, that means it's going to mask over that lowercase t. So in this case, we've got that dominant trait present here, so that means that's going to be tall. That outcome would be tall, and these 2 would be short. So how do we apply that? We've got 2 of them that would be tall, and 2 that would be short. And that aligns up with our genotype that we did. We had 2 capital T, lowercase t, so that lines up with this, and then 2 lowercase t's, which means they're going to be short or recessive. So that's what we did in class that for some reason got chopped off, and the camera didn't record that. So we'll wrap this up and give you your first assignment on Monday when you return, and then we'll have time to work on those on Tuesday the 21st before the Thanksgiving holiday. All right, so that's all we had. We will see you when you come back. Best wishes out there, and all we can do is our best. Catch up to you next time.